hopefully you've had a bit of a break since the first lecture because there was a lot of material there to take in. I just want to follow up by talking a little bit about the specific uses of fMRI as well as some of its limitations. So I just want to jump in because the most common use of fMRI is what we normally refer to as brain mapping. Trying to figure out where in the brain is active when somebody's performing a particular task. And there are good and bad examples of this. One of the really lovely examples was some work that Marty Serino did in 1995. Marty was a visual neuroscientist, and one of the things that was known was that the structural anatomy of the visual cortex doesn't correspond to the functional anatomy that well. And in fact, I mentioned this previously in the Holly Bridge study. So for instance, the calcarean sulcus, which is the site of V1, only part of the calcarean sulcus has V1 in it. And the way you can tell anatomically is if you can see those bands of Gennari. And if you really push the structural MRI to its limits, then you can do that. But in practical terms, that wasn't available in the 1990s. And what Marty Serino discovered was that you could do this functionally. So what he did was he created these stimuli, which are shown here on the left of the screen, where there would be a circle that would start and then expand gradually, and then come back, and then expand gradually. So it was following a very particular pattern where it would start very focal and get larger and larger. And that creates this activity wave in V1 that goes from central to peripheral. And as soon as it comes back, it resets. And that's what he was doing with this. He was taking advantage of the fact that we know that V1 has a central to peripheral organization, and it resets as soon as you get to V2. So independent of whether or not you're in the calcarean sulcus or where in the calcarean sulcus you are, you can see these transitions from V1 to V2 based on these eccentricity maps. The other thing that he did that was very clever was he had these polar angle maps. So that is to say that there was a little piece of like a pie shape, a little wedge, that would be flashing, and it would rotate around the screen in a little clockwise pattern or in a counterclockwise pattern. And again, it's taking advantage of the retinotopy of the visual system. So the fact that you have a spatial layout of your V1, V2, V3, and 4 even, means that by combining these eccentricity maps with these polar angle maps, you can identify where the borders between these regions are. So for instance, what you can see hopefully here, this is, let's start with the polar angle maps. This is the midline of a brain. It's a little bit hard to see all of the occipital cortex this way, partly because some of it's in sulci, which is the depths of the valleys, and partly because the whole visual cortex is not visible on the midline. So what Marty did was he created some software that allowed you to inflate the brain so you could see into the sulci, which are these dark crevices, and then also cut it open and flatten it out. So this is a flat map of the occipital cortex, and hopefully what you can see is there's a dark blue purple alternating with red, alternating with dark blue purple, alternating red, and what's going on is that dark blue purple is that sort of horizontal meridian, the red orange is the vertical meridian, and as you move from purple to red and then reset, you move from V1 to V2 to V3, etc. So this was a functional way to identify in each individual the specific areas of their brain that corresponded to the early visual fields. And this particular method, although it was developed 25 years ago, is still used routinely in vision neuroscience. So this is a really beautiful example of brain mapping. And in fact, there are lots, but this is a nice one. And this is just how you would interpret those blue-red maps. You can see where it goes from upper and lower fields of visual V1 to V2, etc. Now, another thing that people often are interested in is not just identifying what are the brain regions that are involved in the task, but asking questions about the nature of the information processing that's happening in that brain region. And there are lots and lots of ways of doing this. One of the more common ones and one of the more important ones is to take advantage of something called neural adaptation. So this is some work that Bob Desimone's group did in the mid-90s, and this is actually cellular recordings in awake monkeys. So these are probes that are in parts of the extrastriate visual cortex, and what he found were certain neurons in those areas were sensitive to shape. So for instance, if you showed the monkey a triangle shape, you get this kind of characteristic pattern of response. It was interested. You showed it another different shape, like a rectangle or a square, and you got something very, very similar. But as soon as you showed it the same shape that it had seen very recently, you got a reduced response, a reduction in terms of the activity. So it was interested in shapes as long as they kept changing. As soon as you start giving it the same thing, you see this adaptation, a reduction in the activity of the actual cell. And these are spiking rates over time. So these are direct measures of cellular activity in this particular case. Now, as I mentioned, that's not practical in human beings for a lot of reasons, not least of which are ethical. So the question was, can we see something similar to that with fMRI using bold signal? And this is really a nice experiment that sort of used exactly the same kind of approach from Stan Dehaan's group in France, where he was looking at where in the brain does writing expertise exist, right? And what he did was he took advantage of this neural adaptation phenomenon and also a parallel psychological phenomenon called repetition priming. So what he did was he showed people visual noise. You see these shapes that are on the screen. And then in the middle of that visual noise, a word would appear for a very short period of time, 29 milliseconds. And then it would be covered up by more noise. It was so short that people weren't aware consciously that they had seen it. But if the word they saw after that was the same word that they had seen subconsciously, then people are faster and more accurate to respond to that. So that's the psychological effect, the repetition priming. But what's really interesting is you can also do this kind of neural adaptation here, which is to say that if the words are the same, then you would expect less of a response in areas that are interested in the words because they're showing this neural adaptation. And what Dehaan argued was that if the word was the same independent of the case of the letters, so for instance here radio in the prime, the one that's embedded in the noise, is in all uppercase letters, whereas radio, the target word, is in lowercase letters, and other than the I and the O, the letters don't really look that similar. Like an uppercase R and a lowercase r look different from one another, as do A and D. And what he reckoned was that if your brain doesn't care, it recognizes it's an R independent of the case, then that should show a priming effect. That should show this neural adaptation. Whereas if it really does care, then it could be something else. It could be some sort of low-level visual process that has nothing to do with the fact that you recognize the writing itself. And what he found was that there was a part of the left ventral occipital temporal cortex, so an area of tissue on the ventral surface that was at the temporal occipital border, showed this kind of repetition priming reduction, this neural adaptation effect. And it didn't matter whether the words were in the same case or in a different case, this was part of the brain that showed this clear reduction. So if the two words were the same, which are the black bars, you got less bold signal than if the two words were different from one another. So you got a full proper response to both. And this is a really clever way of using this kind of neural adaptation paradigm in order to probe in detail what the nature of the processing in this area is. So based on this, he was concluding that this is part of the brain that's sensitive to your ability to recognize written forms, because it doesn't really care about size and space and case and things like that, just like you don't. As soon as you see the word, all of those features are really irrelevant. What you focus on is the word itself, independent of the way it's been presented. So this is a really clever study, right? I mean, he's used this idea of neural adaptation, combined it with this psychological tool of repetition priming, and he's gotten to the point where he can use the results to argue this piece of cortex, this piece of ventral occipital temporal cortex, is sensitive to the visual structure of words. And in fact, he argued that it's the part in your brain that enables you to read. Now, that's a conclusion that I will argue with a little bit later, but I think that the design of the study was very, very clever and is a really nice example of how you can use fMRI to probe the nature of information processing in a particular part of the brain. The third example that I want to talk about is this idea that brain regions aren't really working by themselves. They're, by their very nature, interacting with one another. And those interactions are a really important part of the information processing that happens in your brain. So this is a really fantastic and engaging study that Biswal did back in the mid-90s. So he did this experiment where he just did something very much like the Peter Bandittini. He got people to tap their fingers and then pause, and then tap their fingers and pause. And one of the things that you see when you do that is you don't just see one part of the brain light up, you see a system of the brain light up. So you can see it here in this image. There are 3 regions, including the primary motor cortex for the hand. Now, as you know, like in this case, I'm tapping my right hand, and of course the brain is contralaterally organized, which means that the left primary motor cortex is what's controlling this hand. Even so, you see activity both in the left and the right. So it's never the case that you just see one or the other. There's some activity in both. But in addition, you also see activity in the area labeled C in this image, which is the supplemental motor area, or the SMA. And again, what's interesting about this is, just by touching my fingers and following my instructions, I'm doing more than just simply moving my hands, right? I'm getting some sensation. I can feel the fingers, so there's not only motor cortex, but there's sensory cortex. But in addition, I'm keeping these basic task instructions in mind and trying to remember what the task is. What finger did I just tap? What's next? Etc. So you also get some certain premotor regions like the SMA involved. So even though it's a very simple task, it activates a system of regions in the brain which are obviously working together. What Biswal did that was super clever was he then got those same people to stop doing the task and just lie there at rest for 5 minutes. So this is a bit like Angulomoso's, completely doing nothing kind of condition. And because it was the same people, he was able to say, look, I know in this participant where their primary motor cortex is because I could see it in the finger tapping condition. If I just go and get that part of the brain while they were lying at rest and say, where are other parts of the brain that are active and over time in the same pattern as this? So even though they're doing nothing, this is what's known as resting state data, because they're not doing an explicit task, they're just resting. He could see that by seeding the brain in the primary motor cortex and saying, look, I know where their primary motor cortex is, what other parts of the brain are going up and down with primary motor cortex. He recreates this whole pattern of left-right primary motor cortex as well as that supplemental motor area. So the system that you see in A on the left of the screen, when people are actually doing a finger tapping task, is recreated in its entirety when people are just at rest. So it's not as strong, the activation, but what you find is that those parts of the brain that tend to be coactive, those parts that work together, also tend to be coactive when they're not doing anything in particular, right? So when they're at rest. And what's fascinating about this correlated activity at rest, is the fact that it seems to create networks of brain regions that work together. And how you weight those regions and how you break them up in different tasks may be one of the sort of fundamental ontological ideas about how the brain actually functions. But the key insight here is that it isn't a region and another region and another region. What we're talking about here are systems that are interacting, and systems can interact in complicated ways. It's not like we're just changing region to system. You can have parts of a system interact with parts of another system, etc. So there's a lot of combinatorics to this as well. And Biswal was the first person to show that you can get this kind of information out of resting state data. And this is what's known as functional connectivity, because these are regions that are connected based on the fact that their activity over time is correlated. Now, there are other ways of using fMRI, and we'll talk about some of them, particularly when we talk about sort of advanced experimental designs. But those are the big three. And I've been very enthusiastic about them because they have enormous amounts of potential. But it's also really important to think about what are the limitations that fMRI has as well. And that's what I want to talk about in this final section. So I'll talk about the spatial resolution, which I've mentioned as a strength, but is also going to be a limitation. I want to talk about something called magnetic susceptibility artifacts, the effects that Lorentz forces have in the scanner. One of the fundamental limitations of fMR is this idea that we can measure correlations, but we can't measure causation. And I'll explain that in more detail, although I'm sure for many of you it's an idea that you've seen before. And then finally, how do we interpret the results? Because we can get these measurements, and that's a really important step, but that doesn't necessarily mean that we interpret them in a way that's consistent between groups of sciences. So interpretation is an important part. So the first thing I want to talk about a little bit is spatiotemporal resolution. And this is a figure that has become super famous. Vince Walsh and Alan Cowie, produced it about 20 years ago in a review paper. And it shows 3 dimensions of resolution. Along the x-axis, we have spatial resolution. And this is on a logarithmic scale. So the largest part is shown on the left. We're talking about like whole brain resolution. And on the right, you get down to the level of like individual molecules, right? And obviously it goes down very rapidly because it's a logarithmic scale. But what's interesting is that imaging techniques like EEG, electroencephalography, magnetoencephalography, and versions thereof, have a spatial resolution that's somewhere between centimeters and whole brain, right? And to the extent that there are times when you cannot accurately tell that a signal in an EEG system has come from either the left hemisphere or the right hemisphere, right? But that's not what it's good at. Spatial resolution isn't the strength of EEG. Whereas if you move a little bit to the right in this image, you get things like fMRI, where you get things that are at the level of centimeters to millimeters. And then if you go even further, then you can get like single cell recording devices and things like that, where you get down to the level of columns or actual single cells, or even single channels or molecules. So that's the x-axis. The y-axis is shown in terms of temporal resolution. So that is to say, it's going to go from millisecond resolution down at the bottom all the way up to like your whole life. And again, it's a logarithmic scale, so it's a very rapid change. And here, EEG, ERP, MEG, that's where they really shine. They have millisecond resolution. You can tell what's happening millisecond by millisecond, which is exactly the time course that most of the interesting things are happening in terms of brain activity anyway. So the fact that you don't have great spatial resolution is more than made up for by the fact that you do have amazing temporal resolution. fMRI, on the other hand, has poor temporal resolution, and that's because of what we already talked about, the fact that the signal that we're measuring is based on blood flow changes, which are reasonably slow, on the order of seconds. So despite the fact the interesting neural changes are happening on the milliseconds, the changes in terms of that we can measure with fMR are much slower than that. So you have some advantage in terms of spatial resolution, but a disadvantage in temporal resolution. And again, more invasive methods like cell clamping or patch clamping or any of those kind of things will give you both spatial and temporal resolution, but they're incredibly intrusive. You need to drill a hole in someone's skull and plant an electrode in them. So the final dimension in Z here is intrusiveness, right? So anything that's coming out of the screen towards you is a correlative measure. EEG, fMRI, positron emission tomography, MEG, etc. They are all correlative and completely non-invasive. On the other hand, anything that's going into the screen is intrusive in the sense that it can be used to change activity in the brain. So not just measure what's happening, but change what's happening and see how that affects behavior. So things like TMS, cooling, and microstimulation are all invasive measures that allow you to influence activity and change behavior. So that's what this picture is showing. So one of the important limitations then of fMR is the temporal resolution, right? So I've suggested that it's pretty good on spatial resolution, particularly for structural imaging. For functional imaging, that's true, but it requires fancier devices. So for instance, the 1.5 and 3T scanners that we have down in Buckney are pretty good up to maybe a 2 by 2 by 2 millimeter resolution. Beyond that, you're losing enough signal that you can't be very confident in it. However, if you go over to the Phil, where they have a 7 Tesla scanner, then you can get down to the individual layers of cortex because there's so much more signal in that you can benefit from the spatial resolution. It doesn't help you at all with temporal resolution because you're still measuring something that's about bold signal, and bold relies on blood flow changes rather than direct measures of neuronal activity. One of the other things that's not always obvious to people is that there are what are called magnetic susceptibility artifacts in MRI. And this, despite the fact that magnetic susceptibility is the basis of the bold signal, that is to say that those hemoglobin atoms that are unprotected by oxygen cause distortion of the signal, which causes less signal. Well, the same thing happens on a larger scale with other structures. So the magnetic field in tissue is pretty homogeneous, right? So in different parts of your tissue, it's pretty solid. But as soon as it moves into a different medium, like air, the magnetic field changes strength. And this is a susceptibility change. So for instance, you've got sinuses behind your nose, you've got a central sinus here, you've got sinuses in your ears, all of which are filled with air, which means that the magnetic field that covers your head when you're in an MRI scanner is more or less homogeneous when it's in tissue, but as soon as it goes to an air-filled cavity, it changes strength. And that causes a distortion of the field, which means that the frequencies that we record are in the wrong place. And you can get two different effects from this. One is what's known as distortion, and you can see that here in this image. I have a structural underneath this image, right? But when you look at the functional slice through that particular plane, there's a hole missing, basically right here. And the reason for that is because there's an air-filled cavity between my skull and the brain tissue that changes the magnetic field, which takes the signal and pushes it back. So it's caused distortion. The material that should be here is now a centimeter or two behind it. And that means that it's been misplaced because of this distortion in the magnetic field. Similarly, there's also something called dropout, which you can see here. And this is the tissue right above the nose. So this is a plane that's about here. There's no sinus at that point in my head. It's just that's above the big nasal sinus. And that's caused a loss of magnetic field, which causes a loss of signal. So that signal in that particular area has dropped out and would have to be recovered. Now, there are ways of dealing with distortion and dropout, but in doing that, you cause more distortion and dropout in other parts in the brain. So it's a fundamental property that you have to deal with fMR. Anytime you do a scan of a human being, of a living human being who's got actual sinuses, then there are parts of the brain that are less well scanned than others. So you might naively think that you put somebody in a brain scanner and of course you measure activity from their whole head, and that's true, you do. It's just that there are certain areas near these air tissue boundaries where the signal is not as good as others. And in fact, in some cases, it's really dreadful, which means that there are parts of the brain that are much harder to image than others. And that will turn out to be important as you're designing your experiments, because if you're interested in one of those areas, then you have to be aware that it's more difficult in fMRI. It's worth pointing out that one of the advantages of positron emission tomography, or PET scanning, that existed and predated fMR, is that you don't have that problem. It's not based on magnets, and as a result, you do get equally good signal throughout the whole brain. So there are times when a positron emission tomography study might be a more effective way of getting at the kind of questions that you want to ask than using fMRI, even though, for the most part, fMRI has supplanted a PET as the imaging tool of choice. This one seems so obvious that it's probably not worth mentioning, but I actually think that sometimes we can sort of miss at the elephant in the room. MRI scanners are loud, and they're really loud because the magnets that are used to create the big magnetic field are always on. In addition, the gradient magnets, the magnets that allow us to encode space by manipulating the field in the three different directions, are magnets in a magnetic field. And magnets within a magnetic field cause sounds. That's what Lorentz forces are. They cause it to physically vibrate, which causes movement in the air, which causes noise. So the way your speaker works, or your earbuds work, is that there are magnets within magnets, and by changing the magnetic fields, you can actually cause it to generate sound. Well, that's exactly what a scanner does as well. There are magnets within magnets that are producing these sounds. The sounds aren't the interesting thing. We need the changes in the magnetic field in order to record the bold, but it has the consequence that it makes a lot of noise. Old scanners produce sounds that were up to sometimes like 100 and 125 decibels. The scanner that we have downstairs, the 1.5, produces about 85 decibels. And the 3T is even louder than that, 90, 95 decibels. The consequence of that is that it's very hard to do acoustic studies in a scanner because it's confounded by the fact the scanner itself is making a lot of noise. Now, there are ways around that, and we will absolutely talk about them in a couple of weeks when we talk about these sort of advanced experimental designs. But it's an important thing to bear in mind. sometimes just the fact that it's a noisy environment changes the way people do your basic cognitive tasks. So you need to be aware of it at all times. I like this slide because it was a real example that was on the front page of the Irish Times a few years ago. They literally had this article that read, "Tree-lined streets can raise children's intelligence." No, they can't. This is a beautiful example of correlation does not mean causation. Why do kids who live on tree-lined streets do better in school? Is it because they're smarter because they grew up with trees outside? Or is it because they live in upper middle class neighborhoods where the schools are really well funded and they've got helicopter parents who devote a lot of time and energy and money to making sure that their kids succeed in school through tutoring and in their own involvement, etc. Right? Those people live in tree-lined neighborhoods. That's not surprising. They're upper middle class. So this link doesn't tell you anything about like the value in planting a tree to make sure that your kid grows up smart in school. That's nonsense. That's mistaking this correlation for a causation. And in fact, this is something that almost every stats class will talk about to some extent. But you have to bear in mind that when we're doing fMRI, along with a lot of other different types of imaging, we are measuring correlations. All we know is that these things that we're measuring are correlated with the behaviors that we're looking for, the tasks that we're running. We do not know that the brain activity that you see is causing any of the activity, in the brain. So for instance, let me give you an example. If I put you in a scanner and just played single written words on the screen and asked you to read them out loud, that'd be easy. You're all good, skilled readers, no problem. But among other things, I'd see a load of different parts of your brain active. I'd see Broca's area active, I'd see Wernicke's area active, I'd see parts of your visual cortex, I'd see that ventral occipital temporal area that Dehaan described as a reading area. But if I look at all of them, do you need all of those things when doing the reading? The argument would be, if you believe in causation, sure, they're all equally important. But that's just not true. So for instance, the reason that you're seeing Wernicke's area and some of the auditory areas is because you can hear your own voice. You can read perfectly well without hearing your own voice. So you don't need that part of your brain. That would be an example of a sort of an epiphenomenon that's due to correlation, but not causation. And I can explain that to you because that makes sense to everybody. But if you're just looking at any given study and saying, well, how do I tell whether this region is needed or not? The answer is you can't. There's no way to tell that from fMR. And that is a fundamental limitation of this correlation is not causation. And then I just want to get to this idea of interpretation, right? Because one of the things that happens, that I've alluded to previously already, is that you can do brain mapping studies that don't make a lot of sense. So I humbly submit to you, where in the brain is chess playing skill is a good example of that. Because what it is essentially a sort of modern form of phrenology. You want to read the bumps on the skull, or in this case the activation patches in the image, and say, oh, that's where chess is in the brain. Well, no. We don't have brains that evolved to play chess. What we have are cognitive systems that are used for tasks like that. So you could potentially rephrase the question and say, well, of the parts of the brain that are active when people are playing chess, which ones are involved in strategic thinking, planning, perception, memory, etc.? All of that would be a totally reasonable questions. And it moves away from just pure brain mapping in that case to sort of asking questions about the nature of the information processing in those regions. It also starts to give you some information about the way those regions are interacting with one another. So it kind of combines all three approaches. But it's certainly possible to just ask bad questions. And you do get this kind of modern phrenology where somebody will just come along and say, well, I want to know where in the brain something is. And then they do a study and they say, oh, there it is. We've discovered where, chess playing is, or homophobia, or whatever the case may be, how to be a political correspondent. That's just bad science. And there's nothing intrinsic about MRI that stops you from doing bad science or that makes you do good science. It is a tool like every other tool, and interpretation is an important aspect. So you have to design your experiments with a good question, a good design, a good analysis, and a good interpretation in order to be on the good science side of that. And that is exactly the type of material that I want to cover this whole term. So I will take you through every aspect of that and try to give you a sense of how you go about doing that so that you can be confident that when you're using the tool for your own questions, you're doing it in a sensible, strategically plausible, and reproducible manner. Okay, well, I hope after those 2 lectures, there are a couple of things that you've taken out of them. One is a basic understanding of how MRI and fMRI work. And as part of that, the fact that fMRI is an indirect rather than a direct measure of neuronal activity. What we're actually measuring is oxygen levels in the blood, and therefore we're getting this hemodynamic response, which indexes neuronal activity. With that in mind, there are a lot of really valuable uses of fMR, including this idea that you can identify systems using brain mapping approaches. You can identify and investigate regional information processing using things like neural adaptation. And we'll talk about that in much more depth. And then you can also use it to characterize the interactions between brain regions, and that is an important aspect. At the same time, there are very strong limitations that you need to bear in mind so that you're using fMR appropriately and not doing, say, for instance, some sort of modern phrenology. After each lecture, there will be a reading list. So there's a chapter that I suggest that you read, as well as information about papers that I've referred to or think that are very valuable for expanding your information for the material that we've covered. I highly recommend the papers, and for those of you who take the time, I think you'll find it very valuable to go and read some of the original articles.